Hi guys, we will talk about a protocol which is preparation of plasmid DNA by alkaline lysis with STS. How will you perform this? How will you prepare the plasmid DNA by alkaline lysis in lab? So plasmid DNA, it can be isolated from small scale bacterial cultures by treatment with alkali and STS. And the resulting preparation, it can be screened by electrophoresis or restriction endonuclease digestion. Then purification by PEG, it can then the plasmid which is used, it is used as a template in DNA sequencing reactions. Okay, so let's start. So the materials used are, first of all, buffers and solutions. What are buffers we require? So these kits are being available. We can buy the kits and we can extract the plasmid, or we can use the following solution. So alkaline lysis solution 1, it has a particular composition. If you want, I can send you the PDF. Okay, so in the kit everything is available, naming as alkaline solution 1 or alkaline solution 2. The components of it will be it is mentioned in the appendix 1 of this book. So then it is alkaline solution 2. Then solution 2 has to be freshly prepared, okay, at room temperature. Okay, then it is solution 3. Then there is antibiotic for plasmid selection, ethanol, which is absolute ethanol. Phenol is to chloroform 1:1. Then there is TE, okay, which contains RNase A. Okay, the media used is LB preferably. So starting with the method. So first of all, preparation of cells. How to prepare 2 ml of rich media, that is LB. Here we are talking about LB media, and containing the appropriate antibiotic. We will take a test tube. Okay, we will take a test tube. In the test tube we will add 2 ml of LB, okay, plus antibiotic, plus our cells, okay, plus our single cell of transformed bacteria. Okay, the cells here are transformed, which we have already transformed. Okay, this is then incubated at 37 degrees centigrade overnight under vigorous shaking. Okay, overnight and under shaking. Got it? You will add LB, antibiotic and cells. So there are some note which we have to keep in mind, that to ensure that the volume of the culture, to ensure that it is properly shaking, so volume of the culture tube is 4 times greater than the volume of the culture. So if the culture is this up to here 2 ml, okay, the volume should be 4 times, at least 4 times. Otherwise it may come out of the culture tube. Okay, otherwise it may come out of the culture tube. Then the tube should be loosely capped. The culture should be incubated with vigorous agitation. Okay, after after the first step, that is incubation in vigorous shaking overnight, pour 1.5 ml of the culture into microcentrifuge tube. Okay, like for example Eppendorf, we will pour 1.5 ml of the culture in Eppendorf. Then centrifuge at maximum speed for 30 seconds at 4 degrees centigrade in a microfuge. Okay, the store the unused portion of the original culture. Like say for example, if the plasmid is not isolated properly, you will not throw this culture. You will take out around 1.5 ml into Eppendorf for plasmid isolation. But the rest of the culture you will not throw. You will store, rest you will store in 4 degree. These things you have to keep in mind. Okay, now when centrifugation is complete, so in this 1.5 ml, what you will do, you will centrifuge at maximum speed for 30 seconds. Okay, so when centrifugation is done, remove the medium by aspiration, leaving the bacterial pellet as dry as possible. So you have to remove supernatant. Okay, what you have done, you have pelleted all the bacterial culture. Now, how you will do it? This can be accomplished by a pipette tip or a Pasteur pipette attached to a vacuum line, or any way. You can just tilt it very carefully, or you can use the pipette. Okay. Then keep the tip as far away from the bacterial pellet as possible as the fluid is withdrawn from the tube. Okay, the pipette tip has to be carefully removed from the place, and this minimizes the risk that pellet will be sucked into the side arm. So these are the precautions which we need to take care of. And the penalty for failing to remove all traces of medium from the bacterial pellet is a preparation of plasmid DNA that is resistant to cleavage by restriction enzyme. If you cannot remove the supernatant very carefully, then there is a chance that it will become resistant to cleavage by restriction enzyme. Okay, because in the supernatant there are cell wall components which will inhibit the action of many restriction enzymes. Okay, so this problem can be avoided by resuspending the bacterial pellet in ice cold STE. Okay, and centrifuging again. Got it? So you have to carefully remove the supernatant after pelleting it down. So this was the third step. The fourth step is resuspend the pellet in 100 microliter of ice cold alkaline lysis solution 1 by vigorous vortexing. Okay, so you have to use the solution 1, alkaline lysis solution 1. What it will do? Let's see. So the bacterial pellet which was there, it will be completely dissolved or dispersed in solution 1. So vortexing with their base touching the vortex. Okay, if this is a vortex machine. Okay, and this is the base of the Eppendorf. Okay, so touching the base of the Eppendorf with the vortex machine, it will increase the rate and efficiency with which bacterial pellets are resuspended. So touch the base with the Eppendorf. Okay, now I'm not reading the second step. Okay, not required. So carefully you have to remove the solution. There is a technique to remove the solution from the pellet. You have to tilt it a little bit, and the tip has to be touched into one portion of the Eppendorf on the surface of the Eppendorf, and you have to suck it in this way. Okay. Then after the first alkaline solution so after the addition of alkaline solution 1, there is addition of alkaline solution 2, okay, to the same Eppendorf. Then again close the tube tightly. Mix the contents by inverting the tube rapidly around 5 to 10 times. This time do not vortex. So first time in alkaline solution 1 when you have added, you have vortexed. Solution 2 when you add, you will not vortex. No vortex. Okay. Then store the tube on ice for some time, so that the entire surface of the tube comes in contact with alkaline lysis 2. Okay. So that entire tube comes in contact with alkaline lysis solution 2. Then add around 150 microliter of ice cold alkaline solution 3. So first was also ice cold. Second was freshly prepared. Second solution was freshly prepared. But third solution is also ice cold. So you have already kept it in 4 degrees centigrade while you are using it. Okay. You have kept it in cold earlier before starting the experiment. Okay. So solution 3 is when added. Then close the tube and disperse solution 3 through viscous bacterial lysate, again by inverting the tube several times. So this is by microcentrifuge, sorry, this is by vortexing. You have mixed by vortexing. Second, by inverting the tubes. I'm just writing invert. Third is also by inverting the tubes. Okay, then store it on ice for 3 to 5 minutes. Here you have not stored into ice. For the second one you have stored in ice. For the third one also you have stored in ice for some time. Okay, now centrifuge this lysate, bacterial lysate, at a maximum speed for 5 minutes. So this time, after solution 3, after keeping on ice, you have to centrifuge again at maximum speed. At maximum speed for how much time? Now this time for 5 minutes. Okay, after this 5 minutes, after it is done, then transfer the supernatant to a fresh tube. Previously we were requiring the pellet, but now we have to take the supernatant. Take supernatant, okay, to a fresh tube. Take supernatant to a fresh tube. Okay, now in some of the cases, so this is about like we have got the plasmid in this case, but there is an optional step that add an equal volume of phenol to chloroform. Okay, mix the organic and aqueous phases by vortexing and then centrifuge the emulsion at maximum speed for 2 minutes. So what is the purpose of adding chloroform is to phenol? Okay, when we add that, there is a separation of different layers. Okay, then we will vortex it. After adding phenol chloroform, we will vortex it. After vortexing, we will centrifuge it at the maximum speed for 2 minutes at 4 degrees centigrade. Then the upper layer which we get, we will take it from in the new tube. So just one step is different. We will add phenol to chloroform in the, which is an optional step, but maximum time we do not do it. So what is the role of the phenol chloroform addition? It is unnecessary. Okay, we eliminate this step maximum of the times, but it was found that elimination of this step results in DNA that is resistant to cleavage by restriction enzyme. Okay, sometimes when this step is not done, it makes the DNA resistant to cleavage. So what is the purpose of chloroform? It removes residual phenol from aqueous phase. So whatever phenol is there in the aqueous phase, it will remove it. Okay, but what is the role of phenol? Phenol, it is slightly soluble in water. Okay, and it can be displaced into organic phase by chloroform. So what is the role of phenol here? So if there is any phenol is there, it will remove it. Okay, it will be displaced into organic phase by chloroform. Okay, but this practice is no longer recommended. So moving to the next step, recovery of the plasmid. So what you have got till now, we have the supernatant in the fresh tube. Okay, now what we will do, precipitate the nucleic acid from the supernatant by adding 2 volumes of ethanol at room temperature. You have to add 2 volume means double of what we have got. Okay, 2x ethanol. Okay, at room temperature, then vortex it. After vortexing, we have to allow the mixture to stand for 2 minutes at room temperature. So this process is done at room temperature. Okay, so after that, collect the precipitated nucleic acid by centrifugation at maximum speed. So we have to take the pure plasmid. What we will do after that, after standing for 2 minutes, after standing or remaining it in that form standing for 2 minutes, what will happen? We have to vortex it to collect the precipitate. Okay, not vortex, we have to centrifuge it. Okay, we have to centrifuge it at 4 degrees centigrade for 5 minutes. Okay, so we have to like keep the things ready while we are proceeding for the next step. While centrifugation has been started, we will keep ourselves ready for the next step so that the time won't be much. Then again remove the supernatant. This time after centrifugation ethanol is there, so remove supernatant. This time also we need the pellet, so remove supernatant. Okay, as we have described earlier by using the pipette. Then invert the tube onto a paper. Okay, invert the tube onto a paper towel to allow the fluid to drain away. Okay, the pellet is stick to the surface, to the base of the Eppendorf. Invert it so that whatever is in the mouth of the Eppendorf will be drained away. Okay, and if any drops are there, then you can remove it with the pipette. If there are any drops, onto the wall of the tube, you can remove with the pipette. Now again remove supernatant, then again add 70% ethanol. Okay, add 1 ml of 70% ethanol to the pellet and invert the closed tube several times. This time we are doing it for washing. Okay, 70% ethanol you will add and invert the tube several times. Invert the tube several times. You can recover the DNA by centrifugation at maximum speed. Centrifuge at maximum speed for 2 minutes at 4 degree. Okay, now this time after that also you have to remove all supernatant. Okay, now the pellet does not sometimes adhere tightly to the tube. So while you are washing the things, while you are washing the pellet, there is a chance that all the supernatant, when you are throwing it out, there is a chance that pellet will also be like washed off. Pellet will also go out with the supernatant. So you have to be careful. It's most of the time it is stick to the base, but sometimes there are the chances that they are not stick to the base of the tube. So you have to be careful that our pellet should not go away with the supernatant. Okay, so remove supernatant and remove any beads of ethanol very carefully. From any side of the tube, if the ethanol remains, you have to remove it very carefully. Then store the open tube at room temperature until the ethanol has evaporated. So sometimes what we do, we put it into the laminar for drying to remove any kind of, to avoid any chances of contamination, or we can just keep the tube open in the room temperature also. This depends on us. If the environment is like contamination free, dust free, we can keep the tubes open and open and tilted. In the tilted position we can keep it, okay, in the room temperature. But if we find that we are working on that area, we can keep the open tubes in the laminar as well, so that there are no traces of ethanol in the tube. Okay, so you have to keep the tubes open to remove all the ethanol for around 5 to 10 minutes. And if the pellet is dried, okay, so if the pellet is very hard, if you have not noticed, if the pellet became very hard, or if you have dried it in incubator under vacuum or desiccator, if you have not taken care of it in a short interval, then it becomes very difficult to dissolve the pellet. Okay, and there are chances that DNA may denature. Okay, so if you are drying it in the room temperature, around 10 to 15 minutes is the maximum time for drying it in the room temperature. Okay, after with if you do not keep it away, if you dry it for more than 10 to 15 minutes at room temperature, then your pellet becomes very hard and cannot be dissolved after that. Okay, so after it is dried, you have to dissolve the nucleic acid in around 50 microliter of TE buffer. So this 50 microliter is not always the same amount we take for elution. It varies from 20 to 50 microliter depending upon how the quantity of pellet. If the pellet size is bigger, you can take 50 microliter, but if the pellet size is very small, you can take 20 to 30 microliter of tea. So tea is already prepared in the lab, which has pH of 8, which contain around 20 microgram per ml DNase free RNase A, okay, which is pancreatic RNase. So vortex the solution after adding tea buffer for vortex it for few seconds and store the DNA solution at minus 20 degrees centigrade. So plasmid we store at minus 20 degrees centigrade. Okay, so this was all about how to isolate the plasmid using alkaline solution 1, 2, 3. So these are all available in the kit. You can also prepare the solution by yourself if you want it. I can share the PDF with you all. Thank you so much.